Gene expression in prokaryotes takes place in a single region, as transcription and translation can happen at the same time. Regulatory proteins are encoded in systems called operons, which are clusters of genes with a common function. The operon is made up of a promoter, an operator, and structural genes. So promoters, this is like the rest of the DNA. So promoters are non-coding sequences where RNA polymerase can bind to and proceed with transcription. And the operator is also a non-coding sequence, and that's where the repressor proteins can bind to. Structural genes are the coding sequences that contain the genetic code for the proteins. So there are two types of operons, inducible and repressible. Inducible operons generally have a function to digest molecules and are only turned on when a specific inducer molecule is present. So one example is the lac operon, which produces the proteins required to break down lactose and has an active repressor, meaning the repressor is already binded to the operator. So when lactose is present, it's first converted into allolactose, which binds the repressor and it makes it inactive. So it looks like deformed now. And since it's like deformed, it can't bind to the operator anymore. And so RNA polymerase can bind to the promoter and just proceed with transcription. But when lactose is absent, then the repressor just remains active and attached to the operator. And this prevents RNA polymerase from binding to the promoter. And since it's like kind of blocking the pathway, transcription nor translation occurs. Repressible operons, they generally have a function to synthesize molecules and are only turned off when a specific co-repressor is present. So one example is the TRP operon, which produces the enzymes needed to synthesize tryptophan, which is an important amino acid. The TRP repressor protein, it can't bind. It's like already deformed. So it can't bind to the operator on its own. So when tryptophan is absent, then the repressor can't bind to the operator, and so RNA polymerase can bind to the promoter and transcribe. But when tryptophan is present, then it binds to the inactive repressor and makes it active. And so since it's active, it's able to bind to the operator, and this prevents the RNA polymerase from binding to the promoter and transcribing. So it's like blocking the pathway. And this basically just saves like valuable resources in the cell when sufficient tryptophan is already present. So in eukaryotes, gene expression can take place it takes place in multiple regions and multiple stages because RNA needs to be processed from pre-mRNA to regular mRNA. So in the first step, chromatins can either activate or repress gene expression depending on how open it is. So there are two types of chromatin, euchromatin and heterochromatin. So this is euchromatin, and it's more loose. So the genes are more easily expressed since transcription factors can more easily bind to it. And heterochromatin is more closely bound and compact, so genes are less easily expressed since transcription factors, it's harder for them to bind here. So genes are organized so that there are promoters, the regulatory sequences. Promoters, regulatory sequences, and the coding sequences. So at this step, the genetic information is pre-mRNA, so the introns haven't been spliced yet. So it's intron, exon, intron, exon, intron. And RNA polymerase can't initiate transcription on its own, and it needs the help of transcription factors. And general transcription factors are proteins that can bind to any promoter complex, and it basically just helps RNA to like bind to the promoter so that it can transcribe. And those are general transcription factors. There are also specific transcription factors, and they only bind to a specific sequence on the promoter of a specific gene, hence the name. So these transcription factors, they bind to sites called regulatory switches. Enhancers are the name for the regions where activator proteins attach in order to turn on gene expression. And silencers are the name for like where repressor proteins attach. So when gene expression is turned on, an activator attaches to the enhancer region and it bends the DNA. So it looks like this. This allows the activator proteins, so like they'd be here, for example. Let's just say they're STF. And it interacts with the transcription factors that are bound to the RNA polymerase. And they bind here. And there are also like mediator proteins in between that basically like exist between the two just to help them bind. And when a gene expression is turned off, repressor proteins, they bind to these enhancer regions. They bind here, and it turns off gene expression, and it blocks transcription. So translation doesn't occur either.